So, right. So, good evening, everyone. Welcome to the special week course on Introduction to Complex Biological Systems. Today, we will try to cover something or a lecture that is important from the perspective of biological systems and that we have and which we were not able to cover in this lecture or the weeks of this lecture. So, something important, something special. Probably you all have heard about it. So, today we will talk about protein ligand interactions. So, that's quite important for different signaling pathways and protein or say protein carbohydrate interactions are effective in so many ways. Okay, so, right, welcome to the class. So, like I said, today's topic is on protein ligand interactions. So, first we will try to talk about protein protein interactions. So, protein protein interactions occurs when two or more proteins binds together. Proteins control and mediate many of the biological activities of the cells by these interactions. And information about the protein protein interactions is important to improve the understanding of different diseases and can provide the basis of new therapeutic approaches, which is very important point because these protein protein interactions becomes the target for development of novel therapeutics, different drugs, be it small molecules or another proteins, and this has been the hotspot for the design of say newly proteins, peptides using AI. So this has been quite a hotspot for drug design and therapeutics. Okay. So the structure of a protein influences its function by determining the other molecules with which it can interact and the consequences of those interactions. So we have a multi-subunit protein like the hemoglobin, core RNA polymerase, small nuclear ribonucleoproteins, and the ribosome. Now, transient protein interactions are strong and irreversible. It readily undergoes changes in the oligomeric state. So, interactions of protein kinases, protein phosphatases, glycosyl transferases, acyl transferases, proteases, etc., with the substrate proteins. Now, proteins for cell growth, cell cycle, metabolic pathways, and signal transduction. Now, these interactions are very, very important in our lives as any disorder in them can lead to fatal diseases like Alzheimer's, the Creutzfeldt-Jacobs disease, things like that. Perhaps the most important known example of a protein-protein interaction is between actin and myosin, which regulates the muscle contraction in our body. Now, examples of these PPIs are the signal transduction, where the activity of the cell is regulated by the extracellular signals, the transport across membranes, so a protein may be carrying another protein. It has importance in cellular metabolism, like in many biosynthetic processes, enzymes interact with others to produce small compounds or other macromolecules. And you have the muscle contraction, like the myosin filaments act as molecular motors, and by binding to actin filaments, uh, enables the filament sliding. Now, there are different types of PPIs. Now, on the basis of their composition, they can be categorized into the homooligomers. That is, these are complexes having only one type of protein subunits, like the PPIs in muscle contraction. And the other one is the heterooligomers, where the two complexes in a macromolecule are, or the complex, are different. So, these PPIs between the cytochrome oxidase and the TRPC3 is an example of these heterooligomers. Now, if we categorize them on the basis of the duration, so we have the stable interactions like the interactions that last for a long duration. So there is a limit or a boundary condition above which we call them as a longer duration interaction, or a stable interaction like the hemoglobin structure, or a transient interaction. So it's quite a fluctuating interaction where one of the proteins comes and binds, it opens up, and it happens only for a short duration of a time, like the muscle contraction. So these have different functions. So all of these are quite important. You need homooligomers, you also need heterooligomers. You need stable interactions, long-term interactions, or also you need these transient interactions, because you do not want the signaling to be switched on for a long time or switched off for a long time. So these have different functions altogether. Now, the effects of protein interaction is that they can alter the kinetic properties of proteins. So protein interactions are one of the common mechanisms to allow for the substrate channeling, can result in the formation of new binding site, can inactivate a protein, can change the specificity of a protein for its substrate. So it's important that we identify the different interactions to understand the extent to which they can take place in a cell and determine the consequences of that interaction. Now, on the basis of their composition, like I was talking about the homo-oligomers, so these are macromolecular complexes made up of one type of protein subunit. So protein subunits assembly is guided by the establishment of non-covalent interactions in the quaternary structure of a particular protein. For example, the protein-protein interactions that we see in a muscle contraction. So there are several enzymes, carrier proteins, transcriptional regulatory factors that carry out the function of homo-oligomers. The heterooligomers have distinct protein subunits interacting in the heterooligomar form, so which are uh, essential to control the several cellular functions. Now, heterooligomar complex, example hemoglobin or the HEB. So, cell signaling events like protein-protein interactions between the cytochrome oxidase and the TRPC3, which is a cation ion exchange channel. Now, on the basis of their bonding, you have the covalent and the non-covalent interactions. So covalent interactions are of the strongest association, like the disulfide bonds or electron sharing. You have the post-translational modifications, ubiquitinylation, sumylation, and the non-covalent, which is established during transient interactions by the combination of weaker bonds. You have the hydrogen bonds, ionic interactions, the hydrophobic bonds, the van der Waals interactions, things like that. So controlled by the weaker forces. Now, what is ubiquitination? So, it plays an important role in the degradation of defective and superfluous proteins, single changed polypeptide. Now, ubiquitination is an enzymatic post translational modification in which a ubiquitin protein is attached to a substrate protein. Now, the steps includes activation, conjugation, and ligation. So, by ubiquitin activating enzymes, ubiquitin conjugating enzymes, and the ubiquitin ligases. That's ubiquitination. Right. So on the basis of their duration of interactions, you have the transient and the stable. Transient means short period of time, stable means longer period of time, or permanent interactions. Like functional or structural roles. Example, cytochrome C are the stable interactions. And if you have a G coupled, G protein coupled receptors that transiently binds to the GIO proteins when they are activated by the extracellular ligands, or there are certain protein-protein interactions happening in the cytochrome in different receptor tyrosine kinases. So where the proteins are interacting with the other proteins to stop a signaling pathway or to trigger a signaling pathway, right? So these have to be transient, because if you have a protein-protein interaction that is stopping, that is triggering a cell signaling pathway, and if it is on for a longer duration of time, then it leads to cell proliferation, cancer, right? So, stable interactions are stabilized by few electrostatic interactions between long side chains with a small contact surface. And in contrast to other cytochrome C complexes, numerous water molecules are found in the long intermolecular span between cytochrome C and the CCO, or the cytochrome C oxidase. Now, the protein domains are the interactions that are only possible due to structural domains within a particular protein. So, how do we define a protein domain? So, it's a conserved part of a given protein sequence and the tertiary structure that can evolve, function, and exist independently of the rest of the protein chain. So, proteins hold structural domains that allow their interaction with and bind to specific sequences on other proteins. Like, for example, the phosphotyrosine containing motifs. So these are the proteins that carry this. So examples for proteins who carry this motif is the activated receptors for the growth factors, the cytokines, and the antigens. And the recognition protein-protein interactions domain have the SH2 domains, the PTB domains that also binds the unphosphorylated peptides. You have the phosphoserine threonine motifs, so it's recognizing protein-protein interaction domain. So you have the 14-3-3 proteins, you have the FHA domains, you have the WW domains, which also binds the unphosphorylated peptides under proline rich. So WD40 repeat domains. Okay. So next up, what we have next up is the acetylation of the lysine residues. So proteins who carry the motif are the histones. So they are the recognizing proteins creates binding site for the bromodomain. Methylation of lysine residues, proteins that carry this motif are the histones and they create binding sites for the chromodomain. So there are other protein-protein interaction domains as well, like for apoptosis you have the DD domain, you have the dead or the death effector domain. These protein-protein interactions have roles in apoptosis. You have the cold shock domain in chromatin formation, remodeling. Okay, then there are some ring motif, ring domains, HEC, F-box in proteolysis. Then there are different other functions for dimerization, the SAM, you have the vesicle trafficking. So there are things like that. So one particular important domain is the SRC homology 2 or the SH2 domain. Sometimes it's SRC homology 3 or the SH3 domain. So their role is in cellular communication. The structure contains, for SH2, the structure contains 2 alpha helices and 7 beta strands. It has high affinity to phosphorylated tyrosine residues. It is known to identify a sequence of 3 to 6 amino acids within a peptide motif, and it represents a larger class of known phosphotyrosine recognition domains. So there are certain identification methods of these protein-protein interactions, like the Yeast-2 hybrid system, where you can have the split ubiquitin system, split lactamase system, split galactoside system, split fluorescent proteins like the green, yellow, red system. So what is the basic idea is that you have to split a system that when reconstitutes can tell, can give you a, can lead to a certain product. So if you take a certain system that when reconstitutes, for example, if I take a split fluorescent protein, so when it is in a split form, it does not show up any color. So I need a, for the screening, I need it to form a color, or I need some kinds of an expression that will tell me, okay, now my two proteins are interacting. So only when they reconstitute, that can only happen through a protein-protein interaction. I design the system in such a manner, then we can tell, okay, so the color is forming means the protein-protein interaction is occurring. For in vitro, you can go for a co-immunoprecipitation, which is the widely used method. X-ray diffraction, biocore, NMR, we can go for an NMR, spectroscopy, fast display. For computational, there are different, nowadays it has widely expanded, so there are so many things that are possible in silico. So methods, there are other methods as well. You can use immunoprecipitation, protein microarrays. There are resonance energy transfer systems, fluorescence spectroscopy, light scattering, ITC, okay, analytical ultracentrifugation, East to hybrid system. Even we can go for a reverse East to hybrid system, okay. Calorimetry, like I was telling, isothermal titration calorimetry, we can go for an anisotropy, okay, surface plasmon resonance, SPR. We can do X-ray crystallography, NMR spectroscopy. So there are so many methods that are widely available to us to find out whether the interaction is happening or not. So maybe we can focus on a few important ones or the widely used ones. We have the East to hybrid system. So it is for testing the physical interactions between the two proteins, first proved in the Saccharomyces cerevisiae, as that's the yeast system, as biological model by Fields and so on. So for here you need a bet, you need a prey, and a reporter gene. So the bet is the protein that's fused to the uh, DBD. So it's some binding domain that's referred to as the bait or the yeast transcription factor, like the GAL4. And then the prey is the protein that is fused to the activation domain. So you have an activation domain and a DNA binding domain. Okay, so that's DBD. So the prey is the activation domain, and the reporter gene is LAGZ reporter for blue-white screening, right? So if you see that if there is no transcription, you get say white, and if there is transcription, that indicates that there is an interaction that is occurring, you get blue, right? So if protein X and Y interact, the DNA binding domain and the activation domain will combine to form a functional transcriptional activator. The activator will then proceed to transcribe the reporter gene that's paired with the promoter. Now for this you need to use two plasmid constructs. One is the bed plasmid that has the say the GAL4 bound to the DNA binding domain, and you have the activation domain bound to the other half of GAL4 or say protein Y. Okay. So the bed plasmid, which is the protein of interest, fused to GAL4 binding domain, and the hunter plasmid GAL4 activation domain. Now, when you express it in a bacterial strain, the selection genes encoding for amino acids such as histidine, leucine, and tryptophan can be used. So you can use auxotropes of these amino acids, so such that when these amino acids are produced, only then the bacteria survives or it dies. The other experiment is the co-immunoprecipitation. So it's a classical technology used for interaction, identification, and validation. So new binding partners, binding affinities, kinetics of the binding, and the function of the target protein can be studied. So you have a cell lysis by non-ionic denaturant, and then you incubate this cell lysate with a particular antibody, and then removal of the unbound proteins by washing them. So the antibody is bound to some agarose beads. So you have first you bind it with say protein X that has interaction with that particular antibody, and then when the lysate, there are other partner proteins to which it binds, and then you can do a Western blot or a mass spectrometry to analyze the what is the complex that it has captured. It's a fishing experiment. So the advantage includes that both the prey and the bed proteins are in their native conformation in the co-immunoprecipitation assay, and the interaction between the bed and the prey proteins happens in vivo with little or no external influence. So there is no external influence of a temperature, pH change, because if it's happening inside the cell, that's constant pH. But the limitation is that low affinity or transient interaction between the proteins may not be uh, detected to that extent. The next one is protein affinity chromatography. It's essentially a sample purification technique used for biological molecules such as proteins. It is a method for separating a mixture of proteins and nucleic acids by specific interactions of those molecules within a component known as ligand, which is immobilized on a support. Now, if a mixture of proteins is passed over the column, one of the proteins binds to the ligand on the basis of specificity and high affinity. The other proteins are washed off and they are not able to bind, like the maltose binding protein, glutathione transferase, the GST, the staphylococcus proteinase. So these are the ones. So this helps you to identify novel protein-protein interactions, characterize the interactions that are already known to occur, different conditions of interactions with the different protein domains, It helps to manipulate the protein-protein interactions in an attempt to understand the biological relevance. Now, if you want to know how mutations affects the protein's interactions with the other proteins, that is also possible. Now there are different protein interactions database that are available in today's date. So those will give you an idea whether that certain protein-protein interaction is known to occur or not. But if you're looking into some novel proteins, some newly designed proteins, because nowadays all the protein information about most of the proteins are available. So if you have designed some new proteins using some computational tools, AI modeling, machine learning, then it will help you to uh, you can submit that interactions in this database, and then it will be updated to a certain extent. So protein-protein interactions have been deployed in yeast methods that are sometimes not suitable for the plant systems. Proteomic approaches are still challenging, and there are certain plant proteomics organizations that are still developing and improving the connections between the plant proteomic researchers and the related fields. Okay. That's about the protein-protein interactions. If you have any questions, you can ask me. Any questions on the protein-protein interactions? Okay, so no questions. Right. So the next part of it is the protein macromolecule interactions. So let's start with the protein carbohydrate interactions that are the intermolecular and intramolecular interactions between proteins and carbohydrate moieties. So we know that carbohydrates are important biopolymers and have variety of functions. So proteins which bind carbohydrates are known as lectins. So compared to the study of protein-protein and protein-DNA interaction, it is relatively recent that scientists get to know the protein-carbohydrate binding. Many of these interactions involve carbohydrates found at a cell surface as part of the membrane, glycoprotein or glycolipid. Now, these interactions can play a role in cellular adhesion and other cellular recognition events. So, intramolecular carbohydrate protein interactions refers to interaction between glycan and polypeptide moieties in glycoproteins or the glycosylated proteins. So if you classify them, it can be classified on the lectins like the C type, P type, I type. Okay, so the C type calcium is required for its activate the binding. For a P type, there are two types. Mannose 6 phosphate can recognize phosphorylated saccharides. One is cation dependent and the other does not require a cation. I type, it's named after immunoglobulin like domain. So sialoadenosine is one type of lectin that binds to the sialic acid. So the biological function is that protein carbohydrate interactions play an important role in biological function like cell adhesion, signal transduction, host pathogen recognition, inflammation, stabilization of the protein structure. Now there are different ways of studying this protein carbohydrate interaction. You can go with the extracrystallography, which gives you the detailed information on the interaction between carbohydrates and the protein. We can go by titration of nosy experiments. That's the nuclear overholzer effect spectroscopy. From there, you get the idea about the specificity, affinity of binding. association constants, and equilibrium thermodynamic parameters. You can also talk about the molecular modeling, use molecular modeling, fluorescence spectroscopy, spectrometry, dual polymerization, interferometry. So glycoconjugates is the general classification of carbohydrates. These are covalently linked with other chemical species such as proteins, peptides, and other compounds. So glycoconjugates consist of many different categories like the glycoproteins, glycopeptides, and the peptidoglycans. They are involved in cell-cell interactions, including cell-cell recognition, in-cell matrix interactions, and in detoxification processes. Fine. So glycoprotein, so the carbohydrate is an oligosaccharide chain glycan that's covalently bonded to the polypeptide side chains of the protein. The sugars in glycoproteins are attached either to the amide nitrogen atom of asparagine termed as N linkage or to the oxygen atom of serine or threonine termed as O linkage. Now, erythropoietin is a glycoprotein hormone present in blood serum that stimulates production of RBC. Now, most blood group antigens are glycoproteins and their specificity is mostly determined by the oligosaccharide present on the surface of RBCs. So there can be O-linked glycoproteins and N-linked glycoproteins. So glycoproteins are proteins that contain covalently attached sugar residues. The hydrophilic and polar characteristics of sugars may change the chemical characteristics of the protein to which they are attached. Now, the addition of sugars is often required for a glycoprotein to function properly and reach its ultimate destination in the cell or the organism. Glycoproteins may serve many functions in the body. Some glycoproteins provide structure, example collagens. Some glycoproteins are involved in immunity, example immunoglobulins such as the IgG. You have the mucins that are secreted into the mucus of the respiratory and the digestive tracts, where the specific mucins may retain water, thus allowing mucus to serve as an effective lubricant. And the specific glycoproteins present on the surface of the RBCs determine blood group type. Now, lectins are a type of protein that can bind to specific carbohydrates. They may bind to a soluble carbohydrate or to a carbohydrate moiety that is part of a glycoprotein or the glycolipid. Lectins usually contain two or more binding sites for carbohydrate units. They typically agglutinate red blood cells, therefore, lectins are also called as hemagglutinin. Now, some of the plant lectins are given over here. You can check them. Now, two important classes of lectins are the C type and the P type. We also studied about the I type, but these two are most important. The C type require calcium ion for binding to a carbohydrate. The calcium ion will link the sugar residue to the lectin. And the P type lectins contains a phosphate group. So the chief function of lectin is to facilitate the cell-cell contact and communication. You have beta carbohydrates on the surface of one cell that binds to the binding site of lectins on another cell, and they communicate. So binding results from the numerous weak interactions which comes together to form a strong interaction. So it's a cumulative effort of the interactions. And lectins contain two or more binding sites for the carbohydrate units. Now, in addition, the carbohydrate binding specificity of a certain lectin is determined by the amino acid residues that binds the carbohydrate. So in lectins, you have the collectins, that's the C type of lectins, which are part of the innate immune system. Their their function is to bind to the oligosaccharides on the surface of the microorganisms. So binding of collectins to microorganisms may trigger the elimination of microorganisms by aggregation, complement activation, opsonization, or the activation of the phagocytosis. You have the selectins, that is also a type of a C type of lectins that plays an important role in the immune system by recruitment of the white blood cells into the inflamed tissue. Now, white blood cells normally do not interact with the endothelium of blood vessels. However, inflammation causes the expression of selectin on the surface of the blood vessel wall. Selective carbohydrate units on the surface of the white blood cells present in flowing blood can interact with the selectin. Now, this interaction results in rolling of the white blood cell on the endothelial cell surface, followed by the stable adhesion and migration of the white blood cells into the inflamed tissue. So the lectin functions stored in seeds function as storage proteins. They also function in defense. So when an invading microbe or insect disrupts the plant's cellular contents, the stored lectin may agglutinate or sicken the pathogen or the predator. Now, ricin is a toxic lectin present in castor seeds, which inhibits the protein synthesis at the ribosomes. Now, legume lectins are uh, generally binds either glucose, mannose, or galactose. They are involved in the attachment of nitrogen-fixing bacteria to the legumes in the protection against the pathogens. Okay. Now we have something that's important. That is, how do we study the interactions? But okay. But there are other interactions as so that's about the protein carbohydrate interaction. The protein DNA interactions also occur for different transcription factors and things like that. What is important is how do we study a protein DNA interaction? For this, we have introduced a technique or an assay known as the electrophoretic mobility shift assay. That's a powerful technique. In short, we call it as the EMSA for detecting specific binding of nucleic acid protein complexes. So over the past 30 years, EMSA has been the go-to assay to investigate the qualitative interactions between nucleic acids and the nucleic acid binding proteins. It is a rapid and sensitive method to detect protein complex with a nucleic acid. So what is EMSA? It is a gel shift assay or a gel mobility shift assay. Sometimes calls it a band shift assay, gel retardation assay. So it is technique to detect whether the protein is attached with a DNA or RNA or not. So as we know, there are many cellular processes where a protein needs to interact with the DNA or RNA, like in the transcription or the translation. So originally utilized broadly in the investigation of sequence-specific DNA binding proteins, such as the transcription factors. So IMSA has been additionally developed to explore DNA protein interactions, RNA protein interactions. So IMSA is a sensitive method used radioisotopes to label nucleic acids and autoradiography. It is possible to use the very low concentrations and small sample volumes. So the principle of IMSA is that the purified protein and the cell crude extract are usually incubated with the 32p isotope labeled DNA or RNA probe, and the complex and the unbound probes are isolated on the non-denaturing polypropylene gel electrophoresis. Now, a mobility shift assay is the electrophoretic separation of a protein DNA or protein RNA mixture on a polyacrylamide or agarose gel for a short period. The speed at which the different molecules move through the gel is determined by their size, charge, and sometimes the shape. So, how it works? So, you have to prepare the cell protein extract, prepare radioactively labeled DNA, and perform the binding reaction. So non-denaturing gel is important because otherwise you cannot observe anything, and then you have to detect the final result. So the protein binding to the unique site on a DNA will form a complex of a protein DNA in equilibrium with the free components. So when there is a strong interaction, you will see that the K is greater than equal to Kd and a distinct band is observed. However, due to dissociation occurs during electrophoresis, a faint smear will also show up between the two bands. Now, if single DNA molecule has multiple binding sites for an individual protein, then there will be multiple complexes formed and we observe multiple bands. So the protocol or the steps that we follow is first we can go for a labeled DNA, and then we can go biotinylation or by fluorescently labeling it. Then we have to make sure that we are preparing the gel and remove the fingerprints to the glass plate by using methanol and dry all the components before we use it. Then we have to prepare the polymerization mixture, pour the mixture into the glass plate and make sure that we avoid any bubble formation. Then we have to pre-run the gel. So pre-electrophoresis is very important for any kind of gel, be it SDS page or an agarose gel or an IMSA gel. So we have to remove the comb and the bottom pressure from the gel and mount it on the electrophoresis apparatus. Then we place glycerol and dye solution in each well to conduct pre-electrophoresis. This is done just to see the gel has formed properly. Running the gel one, it does not break down. Now coming to the sample preparation. So we have to prepare the sample and equilibrate it for 30 minutes at 20 degrees. Um, this can be carried out while the gel is undergoing pre-electrophoresis. So detection of electrophoretic bands on the basis of radioactivity of the samples. So at the end, we remove the gel and the plate assembly from the electrophoretic device, and then we go to visualize the gel bands. In a lab with dim light, placing a film on or phosphorus in an exposure cassette can lead to autoradiography. So if there is a multiple protein binding, you can see that there are two shifts, and that we call as the super shift. So this one is the free DNA that always runs ahead of the protein DNA complex, and when there is two proteins or two DNA that is binding to one protein, you see a shift. So when one DNA is bound to two proteins, you see a super shift. Okay. So how and why titration is done? So as we know that we are going to have DNA interacting with the protein, and for that to happen, we need to have enough DNA and protein in the solution. So the free DNA, and then we start to increase the concentration of the protein. So with lower concentration, you do not see any binding, but as we go on to increase, and increase, you see the number of protein DNA complexes more and the free DNA has reduced. So from here, how do you calculate the KD? So KD is the dissociation constant. So what is the dissociation constant? So dissociation constant KD means half of my DNA has bound to the protein. So if you look at the concentration where you have 50% of protein DNA complex and protein and 50% of the free DNA, that is more or less the KD of that particular interaction. So say if you have, say, starting from zero point 0.1 micromolar to say somewhat 0.2, 0.4, zero point 0.8, then 1.6, 3.2. So you can tell, okay, so the KD value is around 3.2 micromolar. So that's how we calculate the KD. But there is a altogether different equation for it that we need to get that. But approximately it would be around that. Okay, so the applications are that it is used to study the conformational changes in DNA. For example, the IMSA 2D variant with electron microscope is used for the characterization of the conformation. Analysis of cellular extracts for the presence of certain DNA binding proteins, the free and the bound and the free DNA are separated during IMSA. It proves useful for stoichiometric analysis, the number of protein that binds per DNA fragment. It is used to study whether transcription factor is attached to the DNA or not, or the transcription factor attached is targeted or not, and their activity. So the advantages is that it can be used to study a wide range of nucleic acid sizes, structure, as well as wide range of proteins. It is relatively simple method to perform. It is used for qualitative analysis. It proves useful in detection of protein that binds to DNA and cause difficulty in migration of the fragment on the gel. It's highly sensitive method and usually of low cost, so no special equipments are required. So you need just a gel cassette, and the way you are running an SDS ped, you run a non-denaturing gel. And in this technique, it's possible to use both the crude extract protein extracts and the purified recombinant proteins. The EMSA does not provide a straightforward measure of the weights of the proteins, as mobility is influenced by several other methods. It's not an appropriate method for kinetic studies. Dissociation is one of the mm, drawback of EMSA. It occurs during electrophoresis, thus prevents detection. Interacting biomolecules must have different electrophoretic mobilities. So in conclusion, it's used for various purposes such as quantifying the interactions between DNA and proteins. So works on the observation that protein bounded DNA migrates slowly as compared to that of the free DNA, and EMSA is mostly used method for detection of a protein DNA interactions. Okay, so right. Any questions? So if there are no questions, I think okay, we can start with the question and answers in just 2 minutes. So, right. So let us start with the question and answers then. Okay. Which among the following is or are correct about protein-protein interfaces? The interface is 50% polar. It's usually charged. It's richer in aromatic residues. Exhibit both shape and electrostatic complementarity. So what will be the correct answer? It is, so the first statement says it's 50% polar. That is correct because it's interacting with the hydrophilic part and it's intermediate in character, so that's fine. Polar residues in the interface are usually charged. That's not correct. There can be some uncharged polar residues as well, like serine, threonine, tyrosine. Okay, so the protein-protein interface is richer in aromatic residues. That's true for pi stacking, cation pi interactions, which are crucial hotspots, and exhibit both shape and electrostatic complementarity. That is also fine because there is a like a shape complementarity, and then there is positive negative charge interactions. The electrostatics also matter. So 1, 3, and 4. That's option C. Okay. The next one, monoclonal antibodies act as modulators for targeting large protein-protein interface. They are used in screening of recombinants, treatment of many cancer, diagnostic kits. So which one do you think is correct? So this is basically the applications of monoclonal antibodies. So monoclonal antibodies are used in molecular biology, biotechnology to identify and screen for specific proteins produced by recombinant DNA technology like ELISA or Western blotting. Okay. Then it is also used for therapeutic purposes, immunotherapy. They can target specific antigens on cancer cells to induce cell death or blocking growth signals. And for the diagnostic kits, you have um, there were so many rapid diagnostic tests like the pregnancy test, COVID-19 rapid test, and various clinical assays because of their high specificity of the monoclonal antibody. So 1, 2, and 3, that is option A, that is correct. Okay. The next one says which of the following is or are correct about the SRC homology domain. SRC homology domain. So the first statement says it contains is correct. So the first statement is correct because the SRC protein contains two well-known small protein-protein interacting domains like SH2 and the SH3 domains. It's a non-receptor tyrosine kinase is also correct. It's a prototypical member of the SRC family. So unlike receptor tyrosine kinases, it does not have an extracellular ligand binding domain or a transmembrane span. Option 3 is incorrect. It does not include a SH2 or an SH3 domain. So the catalytic domain is a tyrosine kinase domain, not a tryptophan kinase domain. And the fourth one is also correct. So activated SRC signaling pathways are known to regulate the expression and activity of MMP9, which plays a role in extracellular matrix degradation, cell invasion, and metastasis. So 1, 2, and 4 are correct. Next one. Which of the following is not correct about the phosphotyrosine binding domains? So, which is not correct? So, you have to take the incorrect statements over here. So, if you look into the first statement, so while some phosphotyrosine binding domains can bind to non-phosphorylated motifs, the primary known function is binding to sequences containing the phosphotyrosine, typically within a NPX. So that can be any amino acid that's phosphorylated and a Y motif. The next one, so the first one is correct, and then the next one is the insulin receptor substrate 1 is a classic example of a protein that uses PTB domain to bind to phosphorylated juxtamembrane region of the insulin receptor. So that is also correct. The third one says it is situated towards the C terminus. So in many known proteins like the SHC, the IRS1, the PTB domain is located towards the N terminus or the central region of the protein, not specifically the C terminus. So that's incorrect. And the last one says the PTB domain folds with the plextrin homology domains. It consists of seven-stranded beta sandwich, but it is not typically capped by a C terminal alpha helix, not an N terminal one. It's capped by a C terminal alpha helix, not an N terminal one. So option 3 and 4 are incorrect. So the correct option over here is option D. Okay. So there has been some problem with the numbering. Let's not worry about that. So which of the following is or are true about proteoglycans? So so far what we know about proteoglycans, which of the following is or are true? So the first statement, they are present in both extracellular and intracellular matrices. That is true. Only one sulfated glycosaminoglycan chain is covalently connected to the membrane protein. That is not correct, as a single core protein can be substituted with one or many GAC chains. The next one is correct, as most chains are attached to specific serine residue at the core via common tetrasaccharide linker. Okay, and the last one is also incorrect because the heparan sulfate and chondroitin sulfate are types of glycosaminoglycans, the GAGs. So 1 and 3 are correct. 3. Okay, so usually it's 1 and 3, maybe something. Sometimes you consider, you can take option C because in many academic contexts, option 4 is often colloquially accepted as a true as types of proteoglycans. But so option C is a intended answer. Okay, so the next one, oligosaccharide microarrays are used to determine. So what do we use oligosaccharide multi-arrays to determine? It's quite direct. So oligosaccharide microarrays are used to determine the specificity and affinity of carbohydrate binding to lectins. Why? The glycan microarrays are a high throughput technology designed to study the interactions between various carbohydrate structures and the lectins. The next one is glycosphingolipids are the plasma membrane components in which the dash head groups are the dash. So you have to tell whether it's hydrophilic or hydrophobic and whether it is a monosaccharide or an oligosaccharide. So we know, we have studied about it. So the glycosphingolipids are the plasma membrane components in which the hydrophobic, the head groups are hydrophilic in nature and are oligosaccharides, right? The next one, which of the following structural motifs is generally not found in RNA binding proteins? So we have RNA binding proteins. Which of the motif you won't find in a RBP? So the correct one is the helix turn helix motif, which is a major structural motif capable of binding DNA and is primarily found in DNA binding proteins. So we find RRM, double-stranded RM, zinc fingers that interacts with RNA. Okay. The next one says which of HbA1c test is a method to detect the modified form of hemoglobin. So which of the following statement is there true? Is there false about this HbA1c and or that modified form of hemoglobin? So the first statement is true, where HbA1c reflects the average blood glucose level over the past 2 to 3 months. So it's the typical lifespan of a RBC. The second statement is also true, where HbA1c is found when glucose in the blood binds chemically to hemoglobin. And statement 3 is false. The formation of HbA1c occurs through a non-enzymatic chemical reaction, specifically a slow, spontaneous glycation process involving a Skiff's base and Amadori rearrangement, not an enzymatic pathway. Right, so it will be option 3 only. That's option D. Right, now interactions of proteins with nucleic acids are essential for which of the following biological functions? So, right. Interaction of proteins with nucleic acids play an important role in which of the biological processes? You have DNA replication and repair. So, yeah, that's right, because enzymes like DNA polymerase must bind to DNA template to synthesize new strands. So, the first one is right. The RNA processing and translation, that's also true. So, during translation, ribosomes interacts with mRNA and tRNA to build the proteins. So you need them for RNA processing and translation as well. Gene expression. So this process relies on transcription factors. So that are also DNA binding proteins binding to specific DNA sequences to initiate or repress the synthesis of an RNA. And last one is regulation of chromatin structure. Yes, the histone proteins wraps the DNA into nucleosomes. So the modification of these proteins into the action of chromatin remodeling complexes is essential for the changing how tightly DNA is packed, which controls the access for transcription. So all the four options are correct over here. That's option D. Okay, and the next one, and that's the last one, the last question. Okay, so which of the following methods is or are used for the detection of protein nucleic acid interactions? So which of the methods you can use for a protein nucleic acid interaction? We had studied one, but there are other things can be. So what are those? So a pull down assay, yeah, it's possible. It's an in vitro method to determine the physical interactions between a bet protein and a prey protein or a nucleic acid. So pull down assay is possible. IMSA, we studied about it. So it helps to detect the protein, DNA, or RNA interactions, and from the shift we can tell whether there is an interaction or not. The tRNA footprinting assay, so it's techniques like the DNAs or RNAs footprinting identify specific sequence where a protein binds to a nucleic acid. So the protein protects the binding site from cleavage, leaving a footprint in the result fragment pattern on a gel. And the chip or the chromatin immunoprecipitation, it's a powerful method used to identify the locations in the genome where specific proteins are bound to DNA in vivo. So here also all the four options are correct, right? So yeah, that's all. If you have any questions, any doubts from this class, from the previous classes, you can uh, tell, because this is a special session, special class. So I can take the questions from other weeks as well, which I can really, I'm happy to take all the days. Any questions, any doubts? So there will be one more, so I think that will be the last session. That will be on Friday from 6. So the usually the class is on Friday, so that will be the last lecture. And then I guess you have your exam, so um, you just need to go through the lectures and try to revise them well and just execute it in the exam. So, right, I think that would should be good. Okay, I have not uploaded that in YouTube. Okay, so I will upload it and then send. So the NPTEL team will be sharing it with you. I will upload both today and I will be just posting the link so they can send it to you. Okay, that should be fine. Okay, thank you very much. Then have a good day and we will meet on Friday again.